Good evening, everyone. Good evening to Botany Insiders new marathon session for December 2023 examination. Many students were waiting for our course, so we have started off this particular thing and let's wait for the students to join and then we'll start off with our marathon classes. As we discussed in the previous attempt, in your June attempt also, we had a particular, like a complete marathon session for 2 months and more. So over here as well, starting from today, we'll be having almost daily, except for Sundays, we'll be having on daily basis, we'll be having the classes on our YouTube channel. Good evening to all. Hello, everyone. Let everyone join in and then we'll start with the class. Till then, just confirm whether the audio, whether the video is clear to everyone or not. Are you able to check the presentation? I'm absolutely fine, Lucky. How are you? Let the students join in, then we'll start. Okay. Just confirm about the audio and the video regarding the volume, whether it's correct or not, or you want me to increase it or decrease it. Just let me know in the chat section, everyone. Voice is low, not able to hear clearly. Is it working fine now? Everyone, just check. We have 24 students. Very good, very good, excellent. Very good number of students are there in the class right now. Let others also join, then we'll start with the class. Good evening, Arunipa. Good evening, everyone. So, we are starting off with our marathon classes, with our free classes as discussed. Yes, we'll be continuing this free session up till our examination. Don't worry, okay? We'll be continuing this session till our December examination. Don't worry about it. Okay, so let's begin with our class now. Okay, first of all, welcome to Botany Insiders class. If you are new to our channel, don't forget to hit the subscribe button. Okay, in addition, we have our application. Download our application right away. The link is in the description box. And if you want to have a complete detailed version of the courses, we have two main live courses going on. One is a full course and one is a crash course. Okay, you can download the application and get yourself enrolled in the same. And we also have revision booklets available for you all for your December exam. This is the right time for you to order out these things because it will help you in preparing for the examination in the best manner. Right? So, and in addition to that, announcing one more important thing, we are starting off with the PYQ course in which we'll be discussing the previous year question in the best detailed manner possible on our application very soon. So make sure that you are there in the detailed course. We'll be announcing the dates for the launching of this particular course soon. Okay, now let's start off with our class. We have a very good number of students in the class. So let's understand or let's start off with the very first topic, which is related to your cell signaling. Okay, I have taken a target to take up all the signaling pathways in this entire unit. Okay, in this entire segment that we are going to take up on YouTube, we'll be discussing all the possible signaling pathways. In addition to that, if you have problem in any specific topic, you feel like you want me to discuss it over here, you can suggest the topics in the chat section also. Okay, so we'll be discussing about all of the segments in this particular class, right? So make sure that you are participating in the class properly and make sure that you are understanding all the things. As in, when you find any difficulty, you can ask me. Okay. Should we proceed with the topic now? Any doubt, anything anyone want to ask, you can. Otherwise, we'll start off. Okay. Good evening to all the students. Should we proceed? Okay. So let's understand about what cell signaling is. First thing first, this unit is very, very important. Okay. It's a very, very important unit. In addition to that, it's a very interesting unit. If you understand all the processes properly, if you get through the entire mechanism properly, there is no way that you can mark a incorrect answer. Okay. And if you look into the PYQs, you will see almost every year you can find a question in one way or the other. And the most important segment of the entire process is your GPCR, from where almost like every second year we get a question from here, right? Now let's talk about what is cell signaling. Cell signaling specifically means, let's say you have a cell, or before that, what is signaling all about, or why is a signaling required? See, you have cell 1, you have cell 2. How will one cell, first cell, communicate with the other cell? Because our body is made up of millions and millions of cells. And because there need to be a coordination between the cell, because we need to have a proper coordination between the cells, what do we require? We require that these cells should have a communication with each other. How will they communicate? They'll communicate with each other with the help of specific signals. They'll communicate with each other with the help of specific messengers, right? So what are these all about? This is what signaling is. This is what communication is, that how cell 1 and cell 2 are communicating with each other. Okay. Now, how will a cell, like we have understood that, okay, cell 1 and cell 2 are communicating with each other. This is cell signaling. But how will they communicate? What is the pathway of their communication? This is how you have, like, this is your cell 1. This first cell will be releasing some kind of messages. These messages will be in the form of your ligands, right? And this ligand will be transferred to the second cell. Over here, you have specific structures given the name of receptors. They'll be receiving this particular ligand, and as a result of receiving these ligands, what do we have? We have the transduction, the passage of information to the second cell, inside the second cell, and the response is seen. Did you understand this point? How do two cells actually communicate? So you have the first cell releasing some kind of information in terms or in the form of your ligands. These ligands are transferred to the second cell, which is perceived with the help of the receptor on the second cell. The information which is there on your ligand, it is carried for, or it is transferred to your second cell with the help of the various cascade of events, the sequential event that gets passed on to the generation to the particular cell. And now, what do we have? We have the response seen in the entire cell. Okay, so this is what cell signaling is all about. Remember about this particular thing, very, very important. Okay, quickly understand this, everyone. If you find any difficulty, any problem, you can ask. Otherwise, I'll explain you the various mechanisms, the various ways via which the cell signaling occurs. Okay, go through this, everyone, and just let me know in the chat section whether this pointer is clear to you or not. Everyone, quickly read this. Cell 1, cell 2, and then the complete responses. See, now this ligand can have a number of natures. There could be a number of nature of the ligand. It could be a transmitter, it could be a peptide, it could be a transcription factor, it could be any of, like, there are a number of chemical natures possible for this particular ligand. Okay, there are a number of chemical natures available, or which could via which the first cell can provide the information to the second cell. Is it clear to all? Yes or no? Clear, everyone? Participate in the class. 25 for you are there in the class. I want each and everyone to participate so that you can get your doubts cleared. Understood till here? Right? Now, understand this. What is cell signaling all about? It is the communication between the two cells. The cells will be receiving the signal and they'll be responding to the signal from their surroundings, right? Now, the signals can be extracellular, the signals can be intracellular. That is, you have a cell, the cell can provide signal to itself also, and the cell can get a signal from outside the cell also. Okay, so this is the meaning of extracellular or intracellular. Okay, now next thing is the extracellular signaling types. The intracellular signaling is what? That information from the internal environment of the cell is getting processed. Extracellular signaling are of three types. First one is endocrine, paracrine, juxtacrine, autocrine. What is the meaning of each one of them? Let's understand. Before that, ligands nature could be, it could be a hormone, it could be a growth factor, it could be a cytokine, it could be steroid, it could be polypeptide. Okay, so we can have any characteristic of the ligand. Remember this because in CSIR once a question has been asked in terms of your statement. Okay, in the statement wise question this particular thing has been asked, so remember about this particular thing. Now let's understand the four categories: endocrine, paracrine, juxtacrine, autocrine. I am expecting till here the things are clear to you, right? So let's understand about the next segment, which is related to your various types of signaling. Very, very important. Again, understand this properly. I'm telling you, you come to the classes on daily basis, many topics will get cleared. Okay, you will get to understand a number of topics. So make sure that you are coming to the class on daily basis. So we have a number of types of signaling. Okay, understand about the various types of signaling. First one is autocrine. Okay, I'll be showing you the detailed version of the same also. First, understand the entire thing properly. First one is your autocrine signaling. What is autocrine? Auto means self. Okay, auto means self. When one cell is providing the signal to itself to perform any functionality. Okay, this is what autocrine signaling is. And the most common example of your autocrine signaling is that when a cell has to undergo apoptosis, which is known as programmed cell death, at that particular time, the cell will be auto giving signal to itself that come, like the cell need to provide signal to itself from the internal region that you should undergo self-death. Okay, this is autocrine signaling. Then you have paracrine signaling. Paracrine signaling means that when you have a cell and it provides some signals to the cell which is present in near vicinity, which is present in very nearby location to itself. When it is providing the signals in this particular orientation, it is known as your paracrine signal. Okay, autocrine is providing signal to itself. Paracrine is providing signal to the nearby vicinity. This is what paracrine signaling is. Then you have the third one, which is your endocrine. Endocrine signaling is that when you have a cell and it is providing some kind of signals, and this signal need to be transported to a far away location, to a cell which is present at a different orientation, at a very far away site. So it is known as endocrine. Okay, when the two cells, the inducer and your receiver, inducer is the one which is which is providing the signal, inducer is the one, like receiver is the one which will be receiving the signal. Okay, understand about this particular thing. Endocrine, and endocrine is when there is far away or very large distance between the two. Keep a note of this particular thing. Over here, majorly the bloodstream is utilized as a process, or it is utilized as a path of the transportation. Last is your juxtacrine signaling. What is juxtacrine signaling? So juxtacrine signaling specifically means that when you have a cell, let's say this is cell 1, 2, cell 1, 2. This is cell 1, this is cell 2. When the two cells require a physical contact with each other so that the processing of the response can be there, okay? Physical contact between the two cells is required. This is what juxtacrine signaling is. And the most common example of juxtacrine signaling is your delta notch pathway. Okay. The most common example is delta notch pathway. Very, very important. Okay. Whatever we are discussing right now in the class is of utmost importance. Every single year you can find a question. Either in the match the following format, you can get a question in statement wise format, you can get a question in experimental type format also. Okay, so four types: autocrine, paracrine, endocrine, juxtacrine. Autocrine is when the cell is providing signal to itself. Paracrine when the cell is providing signal to a cell which is present in a nearby location. Endocrine is when the two cells which need to participate in the entire communication, they are present far apart. Juxtacrine when they require a physical contact for processing the entire response. So these are the four types of signaling. Very, very important. Okay, quickly go through this. If you find any difficulty, any problem, you can ask. Okay, quickly go through this, everyone. Very important segment. Clear, everyone, till here? Just let me know in the chat section. Make sure you're participating in the class. Done? Very good. Others also participate. 29 of you are there in the class right now. Very good. Okay. So now look over here. So there is endocrine, paracrine, autocrine, juxtacrine. Endocrine is the cell releases the hormone into the bloodstream which travels to a distinctive part, okay, which travels to a distinctive part in the to the target cells in the distinctive part of the body. Example includes your insulin, the growth hormones, and the estrogen. Then you have paracrine. In the paracrine signaling, the cells secrete the signaling molecules that act locally on the nearby cells. The signaling molecules diffuse through an extracellular fluid and they act only on the cells which are present in close proximity. The distance between the two cells is the main way via which we are defining it over here. Okay. Then example over here is neurotransmitters in the nervous system. Then you have autocrine signaling. When the cell releases the chemical signals and it binds to its own receptors, own cell surface receptor, leading to the response within the same cell. This type is involved in growth, development, differentiation, etc. Last is juxtacrine, the one which requires molecules and then they can they have to have a physical contact, a direct contact between the two cells need to be there for understanding the entire mechanism, for getting the entire mechanism on point. This is required. Okay, so this type of signaling is very, very important, majorly in your embryonic development and also in the immune system responses. Clear, everyone, till here? This is the theory portion for the same. You can go through this. Very important. Hi, Subhi. I remember you. Don't worry. I'm just concentrating on the class right now. Be there in the classes on daily basis, everyone. Every day you'll have a class at 8 p.m. Monday to Saturday. Okay. Khushi ma'am will also be taking the classes. I'll be also taking the classes. So make sure that you're there on the classes on daily basis. Every day you'll learn something or the other in the class. Okay. So study well, prepare well over here. See, autocrine is like this is the detailed version. You can go through this afterwards. Exact same thing. Production of the signals, binding to the cell surface receptors, signal transduction, and then the cellular responses are there. Very important. Then you have paracrine, endocrine, juxtacrine. Okay. So I'm expecting till here things are clear to you. Now we'll be discussing about the complete list of the signalings that we have. Okay. We have what are the various signaling pathways that you need to learn, that you need to understand in this entire unit. Very important. I'm telling you, you can find at least one question from this particular unit every year. Okay. Unit in sense of this topic I'm talking about, this signaling pathways which we are discussing right now. Okay. So let's talk about the various signaling pathways that are there, which you have to cover up. If you're starting from your end, that's a very good thing. And if you're facing any difficulty, so we have courses running, we have a crash course, we have a full course, we have revision booklets available. So you can download our app and you can place your orders for the same. Okay. So let's get through the complete list. So very first important signaling pathway is your GPCR. Very important. Okay. G protein coupled receptor is a very important pathway. Following the same, you also have IP3 DAC pathway. In 2021 and in 22, there was a question asked from this particular region. So both the years question were there. Then you have cholera toxin. Very important. How does cholera toxin actually works? What is the mechanism? Which particular regulator ions get disturbed? How does the entire functionality occurs? Very important. Then you have next pathway, RTK, receptor tyrosine kinase, very important. RAS-MAP kinase pathway, again, very important. In 2021, there was a question asked in which CSIR gave you a diagram. A figure was given to you, and that particular you had to find out the various components like A, B, C, D were written down, and you need to write down which will be the component. So it was directly from your RAS-MAP kinase pathway. It was a 2-marker question. Components were asked with the help of a figure. Very simple question it was. It was a 2-marker, and if you would have done that particular, like if you do the RAS-MAP kinase pathway, you can mark it very easily. Okay. Then there's a concept of jack stack. Very important. Very easy as well. Then you have serine threonine kinase pathway. Okay, then we also have the next pathway, nitric oxide signaling, very important. In 2021, there was a question asked in which CSIR asked you that when, like, if I give you a brief about nitric oxide signaling, if I give you a brief about nitric oxide signaling, so there are two type of cells which are participating in the entire signaling, in the entire nitric oxide signaling, I would say. One is your endothelial cells and the second is your smooth muscle cells. Okay. So when you have the movement of nitric oxide from your endothelial cells into your smooth muscle cells, this nitric oxide goes and binds to the guanyl cyclase. Okay. In order to get the process, in order to get the transcription factor activated. Okay, so that your GTP can convert into CGMP and then the further process can be done. So in the CSIR, this particular concept was asked that nitric oxide goes and binds to which of the following. 4 options were there. It was a part B question and you had to find out. So answer was guanyl cyclase. Okay, so you get questions from the in-depth topic as well. Okay, so you have to keep a note of the various facts which are there. You have to keep a note of the various pointers which are there in the entire topic. Okay. In addition to that, we also have a concept of quorum sensing. Let me write it down here. We have a concept of quorum sensing, very important. Okay. Then you have a concept of bacterial chemotactics. These are very minor topics, but CSIR can ask you questions from the same. Okay. Bacterial chemotactics. Yes. Then you have light sensing. How does rod and cone cells in our eyes help in the sensing of the colors, help in the sensing of the light, and how do they actually work as and when we have the entire processing getting done. Then we have a two component signaling pathway. Okay. We also have a two component signaling pathway in which the entire processing need to be understood. So two component histidine signaling pathway, it's one of the component in your plant physiology as well. Okay. So when we discuss about the cytokinin, which is one of the plant hormone, how does it actually get the sensing done? So it is via your two component pathway. So this is how we correlate the unit. This is how we correlate the various segments of the entire syllabus. And if you look into the PYQs, you can understand CSIR is going very, very dynamic in nature. Okay. You need to have that extra edge, you need to have that extra add-on pointers in your preparation to get the entire thing cleared in your mind, right? Because now going in the mainstream way is not helping in out. Okay. Main thing that is required is you need to have a very dynamic approach. But if you do it in a structured manner, nothing is impossible. I'm telling you, if you do in a very, very structured manner, you can qualify the examination. I always say to my students that this examination is not tough. It's your mindset that makes any task tough or easier. Okay. So have the courage in yourself. Get through, like whenever you're starting any unit. Let me just give you one brief also. Whenever you are starting any unit, be it unit number 4, 1, 2, any unit, make the pointer point like very clear in your mind. Write down the topic list. Write down all the topics that need to be covered, like the way we discussed just now. I wrote down all the topics that are important. Now, I am clear in my mind that which are the topics which I have to cover up, right? So make sure that you're doing in the similar pattern. Agar aap, like if you're going ahead with unit number 1, very good. Write down all the pointers of unit number 1. That proteins is important. Then enzyme topic is important. Then your Ramachandran plot is important, etc., etc. Right? So have a very good clarity in your mind that what is the point which you have to cover up. As and when you are clear with the points, make a strategy, get your source verified that okay, these are the sources from where I have to study, and then you are sorted for your entire preparation. Okay. So these are the topics. Go through the entire listing and just let me know which particular topic you want me to cover tomorrow. I'll cover up the same one. I am targeting like according to my schedule. If I tell you, I was thinking of getting done with plant this GPCR in detail, in depth. We'll be discussing every single thing related to GPCR along with the PYQs. So if you want to get this like this entire topic cleared in your mind, just be in the class tomorrow just for 30-35 minutes, and I assure you, you'll be able to solve every single question. Okay. And this is the entire listing of the same. Is everything clear to everyone till this point? Yes or no? Just let me know in the chat section. 33 of you are there. Very good. I want the same crowds to be there, and make sure that you are sharing the same with your friends as well. We have just started off with this particular sessions, and every day we'll be continuing till December examination. Don't worry about it. Okay. Understood everyone till this particular point? Yes or no? Quickly. You can make a list of this entire thing in your notes as well. Okay. Make a list of this entire pointers which you have in your screen right now. You can take a screenshot also. You can write it down after coming to the this recorded portion again. Okay. So this is about for this particular class. I hope all the things are clear to you. Be there in the class on daily basis. Okay. Be there in the class on daily basis. We have a full course and we have a crash course running for you all. So be there in the class. If you wish to enroll and if you wish to have a very structured preparation with us, be there, and you can download our application to get yourself enrolled in the same. Okay, so this is about it. I hope it's clear. Tomorrow's class timings will be same 8 p.m. Be there. You'll get the link on the app. Like, you'll get the link in the community that we have curated for you all. So we have a YouTube community, like a WhatsApp community for the YouTube students who are studying in this particular thing. So I'll be sharing the link over there. Be there in that particular community. Join in that community. It's free of cost. Okay. In addition to that, the app is also free. You can download the app for free and then you can get enrolled in these courses. Okay. All the best. Good luck for your preparation, everyone. It was a lovely session. 33 students, very good crowd. I want everyone to be there in the class on regular basis. Okay. See you all tomorrow. Bye, everyone. All the best.